Hello, my name is Alexis Martinez. I'm going to be talking about the technique referred to as reverse transcription, polymerase chain reaction, or RT-PCR for short. FYI, I will refer to this technique as RT-PCR throughout this presentation. So, RT-PCR is different from standard PCR because it uses mRNA instead of DNA as the starting template. The technique starts off with a sample of mRNA, and this mRNA is purified so that there aren't any contaminants. The enzyme reverse transcriptase converts mRNA to a single-stranded DNA known as cDNA in a process known as reverse transcription. Then DNA polymerase converts this single-stranded DNA to a double-stranded DNA, and now this DNA strand can be used in standard PCR, where it would undergo the steps of denaturation, annulling, and extension. RT-PCR can occur in either one step or two step. One step combines the process of RT, which is reverse transcription, and PCR in a single tube, and it uses specific gene primers. It is ideal for 1 to 2 gene analysis, while two-step separates the process of reverse transcription and PCR. So cDNA synthesis occurs in one tube and then is transferred to another tube in order to be amplified by PCR. It can use random hexamers or specific gene primers, and it's ideal for multiple gene analysis. RT-PCR is important because it can be used to clone cDNA sequence for use in a different experiment or to determine if an mRNA species is present in a sample. So RT-PCR is the most sensitive technique for mRNA detection currently available. It can be used to quantify mRNA levels from much smaller samples compared to northern blot analysis and RNase protection assay. RT-PCR provides opportunities to further study the original RNA species even when they are limited in amount or expressed in low abundance. Common applications of RT-PCR include detection of expressed genes, examination of transcription variants, and generation of cDNA templates for cloning and sequencing. So the research article I found is called Detection of Epithelial Cancer Cells in Peripheral Blood by Reverse Transcription Polymerase Chain Reaction. They performed this experiment because the number of circulatory tumor cells are very hard to detect, and they need a technique that is very specific and sensitive. This experiment method is based on research done from other scientists, where they detected the number of tumor cells of neuroblastoma and of melanoma using RT-PCR. So the key question asked by the authors of the experiment is, could cytokeratin 20 be used as a marker in RT-PCR to be able to detect colon carcinoma metastasis in blood and bone marrow? The experimental design included that they used 3 cell lines: transitional cell carcinoma, derived RT-112, breast adenocarcinoma, MCF7, and colonic adenocarcinoma, HT29, blood and bone marrow samples. And they extracted RNA from the cell lines: normal blood, normal whole blood, normal bone marrow, and spiked blood. Normal blood, use RT-PCR to convert the mRNA to cDNA and then amplify them for cytokeratin 8, cytokeratin 19, or cytokeratin 20. And after performing the RT-PCR, they had analyzed the samples using Argos gel electrophorus and lithium bromide staining. So they wanted to use cytokeratins because they wanted to use cytokeratins as markers because they are expressed in tissue-specific manners by epithelial cells, and they tested for cytokeratin 8, cytokeratin 19, and cytokeratin 20 because cytokeratin 8 and cytokeratin 19 are widely expressed by mucosal epithelial tissues, and cytokeratin 20 are more distinct for cells that derived from the gastrointestinal gastrointestinal area. So the photo that I chose to share is from the article, shows that products of RT-PCR for cytokeratin 8, cytokeratin 19, and cytokeratin 20. So out of the 3 cell lines that they use, the transitional cell carcinoma-derived RT-112 is contains a lot of cytokeratin 8, and breast adenocarcinoma MCF7 contains a lot of cytokeratin 19, and a colonic adenocarcinoma HT29 contains a lot of cytokeratin 20. So therefore, this photo they were just trying to use it as a standard. And in this photo, they show a single band for the number of base pairs for each cytokeratin. So the number of base pairs were identified using gel electrophorese and staining with lithium bromide. This photo was used to help identify the results of the experiment when the products of RT-PCR for cytokeratin 8, cytokeratin 19, and cytokeratin 10 mRNA in the blood and bone marrow samples. It's basically just used as a standard for comparison. The strengths of this article include that they based their technique on RT-PCR protocol used in other research. Their drawbacks are that they didn't include all photos of results to compare to the standard. So their methods were a bit complicated to follow, and it wasn't made certain if cytokeratin 20 could be used as a target gene for the detection of colorectal carcinoma metastatic cells. The question that I have for you is, what will happen if the mRNA sample was not purified? Other than the sample being contaminated, could the RT-PCR process still occur? Thank you for listening. Bye-bye.